Welcome to Data in Biotech, a podcast from Cornai where we explore how companies leverage data to drive innovation in life sciences. Every 2 weeks, we sit down with an expert from the world of biotechnology to understand how they're using data science to solve technical challenges, streamline operations, and further innovation in their business. Today, we sit down with Will Fondrie, Head of Data Science and Engineering at Talus Bio, to explore how machine learning, mass spectrometry, and innovative computational models are transforming drug discovery. We discuss how Talus Bio is targeting transcription factors, once considered out of reach, with scalable, high-impact data science. If you've ever wondered why transcription factors are historically hard to drug, or how mass spectrometry offers high-throughput, unbiased views of protein-DNA interactions, this episode is for you. Here we go.
Will Fondrie, welcome to the Data in Biotech podcast.
Hi, Ross. Thank you for having me out. I appreciate it.
Awesome. Well, just to kick us off, can you give us a brief introduction to your background and what brought you here today?
Yeah, so I am the head of data science and engineering at Talus Bioscience, a Seattle-based startup where we are developing drugs to target transcription factors in a variety of diseases. But my background, kind of to get into that role, is I think kind of interesting. I got my undergraduate degree in chemistry at the University of North Carolina at Chapel Hill. And there I was actually an undergrad researcher in a physical chemistry lab, which was very different than what I do now. But one of the things when I was looking at grad schools was looking into going into like genomics and things like that. And my PI at the time was like, won't you miss the big machines that we work with here when you do that? And so he ended up being right. When I did my PhD in molecular medicine at the University of Maryland, Baltimore, where I ended up in a proteomics lab where we did a lot of work with exosomes and characterizing biomarkers in exosomes, and then also just a lot of other random proteomics projects and non-proteomics mass spectrometry projects as well. But during my graduate school career, I kind of started out in the wet lab and doing a lot of wet lab work, which I think it's been beneficial, but it turns out I'm not very good at. So most of my wet lab experiments failed in one way or another. But one of the things that all the labs that I was a part of in my time there needed was somebody to do computational work and to develop the analysis strategies that we needed to make sense of all the data we were acquiring. So that's kind of where I naturally fell in, and I ended up loving that aspect of it. And then at that time, I feel like machine learning was on its rise again and became something I got really interested in and started kind of teaching myself on the side. And so when I was done with my PhD work, I ended up going to the University of Washington and working with Bill Noble and the Department of Genome Sciences there, really developing machine learning methods and AI methods for proteomics specifically. So both analyzing the raw data and then making sense of all the data that we get out of that. And actually, that's where I met the founders of Talus. So Lindsay Pino was a graduate student in Bill Noble's lab at the time. She's one of the co-founders. And then Alex Federation was the other co-founder. He was a postdoc in a lab just next door. And so I got to know them during my time there. And when they were starting the company, they reached out to me and thought I'd be a good fit. And I decided to join and haven't looked back since. It's been a lot of fun so far.
Awesome. Well, that's a wonderful backstory for you and also for Talus. Can you tell us a little bit about Talus and what you're working on and the problem that you're trying to solve?
Absolutely. So at Talus, we've built a proteomics-based platform to let us develop drugs against transcription factors. So we're trying to develop small molecule drugs that target this class of proteins that binds DNA and causes a whole host of downstream effects on the cell. And so in disease, these are master regulators of all sorts of genes and processes. And so in disease, they tend to turn on processes that shouldn't be turned on or turn off processes that should be turned on. In cancer specifically, they tend to enhance cell growth and proliferation or to turn that off in the normal cell. And so what we're trying to do is develop these small molecule inhibitors for transcription factors. They've been known to be good drug targets for a long time, but they're traditionally very difficult to develop drugs against. That's for a wide variety of reasons. One of them is that they tend to have these really disordered structures, at least from what we can capture or what we know now. They have these highly disordered structures that are not captured well. They're hard to crystallize, except for their DNA binding domains, which tend to be not great places to develop drugs against because of their high homology across proteins. So you end up hitting a lot of targets when you try and target a DNA binding domain. And so traditional structure-based drug discovery approaches have not really been effective for them. They're also really hard to measure in a test tube in isolation. So they're difficult to purify. And then once you've purified them, they really need like all the components of the nucleus, all the DNA and their cofactors to be active in their normal state. And so these assays that take place, in a test tube with a purified protein, while they can be effective, have traditionally not been very effective for transcription factors for that reason. So our platform leverages the strengths of proteomics to do this in a high throughput way, where we perturb a live cell with a drug or a small molecule compound, and then we go in and we isolate the nucleus and measure what proteins are bound to chromatin and how much of each of those proteins are bound to chromatin at any given time. So we get a snapshot of that cellular state after it's been perturbed. And what that lets us do is infer when we see certain proteins falling off of chromatin, we can infer the target of the drug that we've treated with, or the small molecule that we've treated with, and work to develop those to be more selective and more potent for the targets we care about in disease.
Awesome. So that's really really interesting. I just want to make sure I sort of understand it. Can you dive into why mass spectrometry as an assay is sort of the best way of taking that snapshot of the cellular state when it's been perturbed, and sort of understanding how these transcription factors are being upregulated or downregulated.
So one of the things I love about mass spectrometry is that we get a fairly unbiased view of the system that we're trying to measure. And so in our case, we get a high throughput, unbiased view of what proteins are bound to chromatin at any time. And so that means that we can measure the abundance of thousands of proteins that are either directly bound to DNA or bound through a cofactor to DNA in a single assay. And so we're very high throughput on the range of proteins or the range of targets we can measure, and then kind of medium throughput on the compound perturbation side. And so what that lets us do is, instead of just taking a single target and throwing a bunch of compounds against a single target and seeing what works there, and then having to consider selectivity down the road, we get to measure both the on-target specificity of a compound and any of the off-target effects that we can measure in the nucleus simultaneously. And so mass spectrometry is the technology that we've chosen to let us do this. There are a lot of exciting new, I think, proteomics technologies that are coming online, but they're just really not to the depth and reliability that we need for these particular assays. And then I guess the other side strength that mass spectrometry also gives us is that when we're looking at small molecules that have a covalent mechanism of action, meaning that when they react, they physically react with a protein and will stick to them. One of the things we can do with the mass spectrometer that is very difficult to do with other technologies, or impossible, is we can actually detect where that small molecule is bound in some cases. So we can both get confirmation that we're engaging the target of interest. We can measure the functional effects of that binding event on the proteins binding to DNA, and we can measure thousands of proteins at the same time. So together, those make it a really powerful technology for characterizing what these small molecules are doing to proteins binding DNA.
Awesome, thank you. And I think that serves as a really great platform and background for talking about sort of the computational approaches that you're using at Talus. So, one of the things that I've heard you talk about previously was that you had this sort of recommender system approach to drug discovery. Can you, sort of explain what it means to have a recommender system, and how that relates to the data that you're collecting and sort of the feedback loop that you're creating between the wet lab and your computational work?
Yeah, absolutely. So I'm sure one of the problems that your listeners are familiar with, is that the space of pharmacologically active small molecules is just absolutely massive, so massive that there's no way we could possibly measure every single molecule we would want to measure, or find that needle in the haystack just randomly searching that space. And so what we really need, especially with a technology that's kind of medium throughput on the compound side, is to have some system to prioritize what compounds we should measure first, which ones are going to be the most likely or most informative for us to measure to hit a particular target. And so for that, the way that we've decided to tackle that problem is by first we've kind of developed our own chemical space that we want to search within. But then within that, we've developed our models to recommend which compound is most likely to perturb a transcription factor. And this kind of goes back to the Netflix challenge, which I forget what year that happened in, but the idea was that Netflix has this huge problem where they wanted to, or they need to, given a list of users and a list of movies or TV shows, they need to figure out and predict how well a user will like a particular TV show or movie. And so you can think about this as a giant matrix where one axis has the users, one axis has the movies, and the elements of that matrix are some rating of how well a user will like it. And so the way that Netflix, or the way that folks have solved the Netflix challenge were a variety of these different matrix factorization methods to learn something about what represents a user, and learn something about what represents a movie, and then combine those in different ways to get that predicted rating for each user movie pair. And then Netflix, when you go to browse movies, it can sort that list by your personalized rankings and give you the things that you'll binge watch for days. The way that we're thinking about this is pretty similar. So what we've done is we have a slightly different data structure where or at least how I think about it, we have kind of cell types as one axis of this data matrix, or tensor, as we'll see. We have the protein targets that we could potentially that we measure and could potentially target as another axis, and then we actually have the small molecule compounds as another axis. So this looks like a giant cube if you were to visualize it, and like each box in that cube, each element in that cube, would be a measurement of whether that compound affects the DNA binding activity of that protein in a particular cell type. And so our goal then is to build models that can predict the elements of that matrix, or that tensor, and help us to rank those for particular targets, so that we're measuring the compounds that are most likely to work first. We're trying to prioritize those compounds first. So the way we've done that is we do what's called a deep tensor factorization to make that happen. And so one of the things that I think has been very exciting over the last few years has been the development of these foundation models for representing really biological sequences in a lot of ways. So we have all of the DNA foundation models are very cool, but one of the ones that we use is for protein sequences is the evolutionary scale model. So we're currently using ESM2 to represent these protein targets. And what that does is that gives us a starting point for what does it mean to be a given protein.
So you've got the protein types that you're going to target, and you've got the small molecule compounds that you're targeting them with, and then you've got the different cell types in which the small molecule and protein interactions are going to occur. So can you help me understand, like, what role are the cell types playing in your exploration of what the drug is that you're trying to discover?
When we're thinking about cell types, one of the things we're always thinking about is what diseases is a particular transcription factor relevant in? What indications is it going to be? Is it going to be active, first of all? And is it going to be effective to develop a small molecule against? In different cellular contexts, transcription factors will actually complex with different cofactors and bind different regions of DNA. And so we want to make sure that when we're developing an inhibitor or a stabilizer for a transcription factor, but mostly inhibitors, but if we're developing an inhibitor for a particular target, that it's inhibiting that target in the correct context and not in the context that would cause wider ranging effects than curing the disease that we're targeting. And so that's really what the cell type axis on that represents. And so we're trying to make sure that as we are building out programs and we're taking on new targets of interest, that we're really making sure we're measuring cell types and onboarding cell types that are representative of that underlying disease biology to represent that well and maximize our success in the kind of downstream drug development activities as well.
Yeah, interesting. So that's it. So if you're trying to, sort of inhibit a transcription factor that has implications for a disease of the lung, you want to make sure that it's not having effects on that transcription factor in other organs of the body, like the liver or something like that. Am I thinking about that right? Or are these cell types sort of
Exactly.
different? Yeah.
No, exactly. So that's one aspect of it. The other aspect is like a lot of these cell types have specific subsets of transcription factors that they actually express, especially when you start looking at rare diseases. For example, our lead program is in a disease called chordoma, and there's a transcription factor, the brachyury protein, which is the TBXT gene. That one is often turned on, and it's something that is normally only expressed during development. And so the opportunity to measure that protein in an environment that matters means that we have to go to chordoma cell lines, or there are some other cancer types that have that expressed as well. But we really have to make sure that we're targeting that specific cellular context to make sure that we're measuring some of the things that we care about.
That makes sense. That makes sense. And then, so you've got the example you gave from Netflix was like users and movies, right? So in order to do a really good recommendation, you need to understand a lot about the users and a lot about the movies. And so in your case, you need to understand a lot about the small molecule that you're using to do the inhibition or the stabilization, and then you also need to understand a lot about the transcription factor that you're trying to bind to. Am I thinking about that right?
Absolutely, yeah.
Yeah. So my understanding is that this is where a model like ESM2 comes in, right? Where there's been all of this various data sources that have been fed into it to give it as much information about the proteins as you possibly can. So am I thinking about that right? Or how does DSM2 give you the information you need in order to make the recommendations that are coming out of the system as good as they can possibly be?
Yeah, so you hit the nail on the head there. The naive approach we could take to building this recommender system would be to try and learn representations for drugs, for proteins, and for cell types, all de novo from the data that we've collected. And the biggest downside to that is just that, as in most biological domains, we are data limited ultimately. We can't train massive models from solely our data. And so these foundation models like ESM2 give us the opportunity to inject all of that prior knowledge without having to learn it directly from our data or to bring in those sources ourselves. And so what that looks like for us is that we embed all of our protein target sequences into their ESM2 representations. And so those embeddings are what we're using to represent each protein. We're using Multivac currently for our compounds. And so those give us a good starting point for what does it mean to be a particular molecule. And so given those two starting points, we can then feed both of these through a neural network, and we can learn about cell types de novo and combine all of those three to get our prediction to fill in that element in that cube tensor that I described.
Right. And then the element that's being filled in is sort of some measure of transcription factor binding, or how are you what's the value that goes in that little box, or the little square in this case?
Yeah, so the readout of our platform, which we call TF-scan, in case I mentioned that again, the readout of TF-scan is an abundance of protein on chromatin, and so we can look at we transform that into essentially a percent inhibition or percent gain on chromatin if a protein stabilized. And so that transformation lets us look at the relative change of a compound to whatever basal level of binding that a transcription factor has in a cell type. And so that's what we're predicting. That's what we're imputing into that matrix. And so then we can rank compounds for a protein target by that prediction and look for the compounds that would perturb a given target of interest the most, and measure those first.
So you've been given this list of compounds that are likely to perturb a transcription factor of interest the most. What do you do other than running another experiment? How do you use that information in order to understand what's happening in the biological system that you're studying more effectively?
Yeah, yeah. One of the things we think about is when we start to get into the chemistry of these compounds. Um, we look for similarities in the structures of those compounds. We want to look for patterns that emerge there. You can do like some fairly basic clustering to get clusters of compounds that come out of these predictions that are predicted to be highly effective against the target. But when we're actually trying to narrow down, say, this list of 1,000 compounds to a smaller subset that we might want to test quickly, one of the things we try and do is once we have these predictions is try and just make sure we're selecting for some structural diversity in those predictions. So we get like our list of 1,000, and then we use a submodular optimization process to select what's the best representative subset of these compounds to actually physically test in the lab. And the idea is that hopefully by testing around that space, probing around that space, we're getting much more information than if we took like the top 10 and they were all, very similar to one another in our chemical space.
So, selecting a representative subset of the structural diversity of the compounds that are predicted to be really good is your way of like maximizing information gain from the new experiments that you're running. So that's, creating the best balance you can between explore and exploit. Because,
Exactly.
yeah, interesting.
I guess one of the things that I just want to mention is just that the recommender systems that we're learning are not static. Is that we're constantly integrating data that we've collected based on previous predictions to build better models and in kind of an active learning loop. And I think that's the real key to making progress on particular targets is making sure that you're reintegrating data that you're collecting to maximize what the model can learn.
Awesome. So, in order to you're integrating with more than one model and or bringing in data from all of these different sources in order to add information to the system that can be used to make better recommendations. Can you just give us an orientation to what your data stack looks like in order to manage all of those moving pieces?
Yeah. So a lot of our a lot of so when we think about our data stack, we're built. If we start at the kind of the cloud foundations, we're built on top of AWS. Pretty much everything we do is processed and built in AWS. And so so our raw data we automatically upload from our instruments. It's dumped on S3 in an organized kind of data lake there. We then have to run our bioinformatics pipelines to even begin to work with the mass spec data that we've collected. So our TF scan experiments come out as these raw mass spec files that you then have to interpret to get these protein quantities that are useful for biology. And so for those bioinformatic pipelines, we've traditionally leaned on Nextflow a lot to build those. So we've been using Nextflow orchestrating AWS batch to make that happen pretty effectively. Uh, we contribute to one of the NF Core pipelines. So NF Core is one of the great parts of Nextflow, I think, where there's a lot of community support around these open source pipelines for a variety of different bioinformatics tasks. One of those is for proteomics, or there are multiple for proteomics, but we contribute to one of those to make it scale better and to make it work for our use case a lot. And so we run our data through that as part of our bioinformatics processing. So that's like a vast majority of the compute that we actually have to do is these bioinformatics tasks. We've also recently been using Redone from In-Citro, which is their open source orchestration platform. It's been a lot of fun to use that one and to be back in orchestrating using Python instead of Groovy for the programming language. But yeah, that's what we use to orchestrate between our bioinformatics pipelines and then going into model training and updating, which is what we need to do on a regular basis. And so that's what kind of connects all those pieces together, and it's the plumbing that make it all happen smoothly.
Awesome. And you mentioned bringing, so you've got the data that you're generating from the mass spec machines, and then you also mentioned sort of bringing in external data sources. Can you just orient us toward what are some of the external data sources that you're bringing in, and how you're using them in the context of that stack?
Yeah, so we're not actually bringing in that many external data sources, to be honest. So the most of the external data really gets injected through the model embeddings that we're using here. We are using uh, like protein sequences from UniProt, for instance, for our bioinformatics workflows, and for the embed to know which proteins we need to embed using ESM2. But a vast majority of the external data that we're bringing in for these models comes from uh, the injection from these foundation models that we're using. Um, and so I think one of the things that's I think kind of interesting about that is just like making sure that you the I guess managing the feature store and making sure that as you're updating models using different models, there's all kinds of foundation models you can pull from and use, making sure that uh, you have systems in place to know which ones are the most effective and should be used in the production models, but b making sure you're consistent about how those are updated and maintained and everything as well.
Right. So, I mean, obviously, that's also a challenge with sort of experimenting with different foundation models, and bringing different embeddings into your ecosystem, knowing which ones are the production ready embeddings, which ones are the ones that you're experimenting with, which are the old versions of those embeddings that you might need to roll back to, or something along those lines. Um,
I feel like our young team has really just started to dive into all the fun and all the headaches of ML ops in engineering there. So
for sure. So, relatedly, what lessons have you learned about, designing scalable bioinformatics and drug discovery pipelines based on, what you've been able to build?
one of the things that I think is perhaps obvious to folks who have been doing it for a long time, but it's not necessarily obvious to me getting into it, was just that things unexpectedly break at scale all the time. Even on AWS, like if you're running tens of thousands of jobs at the same time, you'll have an EBS volume fail, and you have to have systems to account for that. So we've gone through kind of the painful iterations of going from these, when we're at the scale of like analyzing, like hundreds of TF scan experiments, it looks much different than the scale that we're at now of analyzing tens of thousands of uh, TF scan TF scan experiments simultaneously. And so being able to handle that data and then trade off kind of the practical aspects of how those pipelines should be run with like the kind of, I guess, the theoretical best practices for how some things should happen, I think is kind of tricky. The other thing has been kind of the the relationship of building software versus buying pre-made solutions. I think um, I tend to lean heavily toward the build side of things, but one of my own flaws has been to kind of realize that in me, and to uh, remember that time at a startup is your absolutely most valuable resource. So when you can pick something off the shelf, it's often worth doing as opposed to re-implementing something yourself.
Yeah, I want to ask you a couple follow-ups on that. So you mentioned it. You mentioned first that so like operating at the scale of tens of thousands of TF scan experiments simultaneously is sort of the scale where things like hardware just starts to break. So you need to have sort of the reliability and the recoverability. Is that are those some of the contributions that you're making back toward NF Core to make it handle those kinds of things, or where like how where in your pipeline do you sort of need to build in that reliability?
Yeah, so part of our contributions to NF Core were to help it scale to kind of the thousands of mass spec runs. We're specifically contributing to the quantum S pipeline in case anybody is interested in that. But we definitely have used that up to the kind of thousands of runs threshold that we were using. But when we really hit, I don't know, 7,000 or so TF-scan experiments, that's when that started to break down. And there's really just kind of one step that is, this might be going a little into the weeds, but there's one step of this kind of traditional proteomics pipeline where essentially all the data has to be aggregated onto a single node, um, where that's just not practical. That becomes impractical eventually. And that's where things really start breaking. And so we kind of designed our own internal pipelines to avoid that step using some heuristic approximations of what's theoretically the right thing to do. And I think that actually has helped us to really break through that barrier and allow us to scale further. But a lot of the optimizations and things that we made to make that happen are really directly tied to the infrastructure we've deployed. So uh, it's one of those things that it could be open source, but I don't think it would be useful necessarily for for it to be open source.
Unless people are replicating exactly the workflow that you're undergoing. Yeah,
Exactly.
that makes perfect sense. And then I know that you historically had outsourced some of your data modeling and warehousing, but later brought it back in house. And maybe like that experience is part of why you have this this bias toward building. Can you just give us an understanding of what what that experience was like, and what that's taught you about the build versus buy trade-off, even in the context of a startup where time is so critical?
So I guess just to introduce everybody to kind of our metadata storage strategy, we wanted to track all of the metadata associated with all of these TS-scan experiments. So we wanted to track, given a TS-scan experiment, we wanted to be able to know what batch of compound was used to treat those cells, what cells they were, where they arrive from, all of the provenance information for that, because it's important for auditability. It's important for downstream downstream tasks in modeling and everything to be able to know exactly what an experiment was. And so early on, we had done kind of built a data model or theorized the data model for what this could look like in a database and in a relational database, and had worked to try and get it implemented through some third party vendors and everything. And when we thought we had it implemented, it ended up being very rough. So we would try and query the data in the expected patterns that we thought it should work in, and then we would either either the vendor the API from the vendor wouldn't exist, or we would need to have some like pre-knowledge of UUIDs to specifically retrieve the things we were looking for, when that was the entire point of the query, was not to use those. Uh, and so we made the tough decision to bring that data modeling and warehousing back in-house, where we essentially have now we have like a relational database um, with all of that metadata stored there, and then linked out to where all the raw data and experimental results live to that. The That transition was very painful, in part because there were other kinds of data modeling decisions that had been outsourced in that process, where there were just nightmares lurking in the database. Like, unique key constraints were enforced, but like some of the unique keys were the same key with a new line character, and things like that. Things where you shouldn't that should never exist in your database to begin with. And so so we went through the painful process of having to extract all that data, clean it, and then uh, rebuild our schemas on top of that to actually manage it ourselves. I think the biggest lesson I learned from that is just to, like, when you're first off, I think you want to think about your data model early on, and know exactly what you want to capture in that, what purpose you want it to serve. Because if you start too broad, you start to try and capture too many things and all the relationships, you add a lot of complexity there. If you start too narrow, it's very painful to go back in time and add things on. So really think about what you want your data model to do, and to specify that um, correctly, and then to be involved in that data modeling process and verify at each step of the way, if you're using an external vendor, that it is actually functioning as you expect it to, rather than going through this painful transitioning and then finding out in the end that things are just not working reliably. So um, yeah, so that's kind of our story about how we had to go undergo this painful process of repatriating that data into our systems. And but I will say now it enables us um, to do things at scale that would have previously been impossible for us. So uh, all of our internal systems now reference it, and can use that data in a variety of ways.
Yeah, that's interesting, and it just sort of highlights the extent to which external vendors will not always have the same quality standards that you have for the way that your data is architected, and also the same sense of what the long term strategic implications are of the decisions that you're making. And so even if you're trading off or outsourcing the writing of the code, sort of like the rigor with which that process needs to be managed and the attention paid to it is like critical to making sure that it establishes a solid foundation for you.
Exactly. And just to be clear, I'm not saying people should never work with an external vendor for their data modeling and everything. Like, you absolutely should just be involved in the process, be more involved than I was, and don't make the mistakes I made.
That makes sense. I know that you're passionate about open source scientific software. So can you just help everyone understand from your perspective why open source software is so important for progress in science?
I think for science in particular, open source is really the only way to make progress as a whole. So when we look at uh, science in general, one of the things we think about is when we make great claims or great discoveries, we have to present the evidence to back that up and present how uh, how something could be. And so when we think about a wet lab experiment, we have to have the evidence to back up the claims that we make about that experiment. And I think the same thing is true for the computational tools and resources that we use. A lot of times in modern times, like a lot of the really big discoveries uh, when we think about like protein folding and other types of challenges that AI has really come in to solve, a lot of those are really cool scientific advances. But uh, if we don't have the code, which is kind of the proof that it is doing what it says it's doing and describes exactly how it's done, if we don't have the code to back that up, then we're really just advertising about a cool new technology at a company or a research group. And so I think the open source is really what allows folks to both integrate that new advance into their own workflow. It lets them test it and verify that it actually does what it claims to do, make sure the claims are verifiable. And then I think more importantly, it allows people to build off of it and to create new things and to make new discoveries and to expand science faster. So I think those are the big reasons why open source is just so critical for just science in general. And then I guess there's also the reproducibility side of things as well, where without the code used to analyze data, or without the code used to for a model, it's impossible to reproduce the results that are presented in a paper. And we already live in a time where it's very difficult to reproduce experiments anyway, and so not having the code there just makes it orders of magnitude more difficult.
Right. Yeah. I mean, and obviously, if it's wonderful if you're able to speak to the exciting results that you're producing, but if nobody else can reproduce those results, then it's an advertisement for your organization, but not necessarily not necessarily a validation of the capabilities that you've claimed to create. Right.
Exactly. And that's not to say there's no place for closed source software. I absolutely think there are places for closed source software. I just think that if you're conflating the closed source software with the scientific advance, then you have just a huge kind of conflict of interest there that needs to be resolved. And I think like those critical pieces of open source scientific software can be the foundation of great companies, uh, that build on top of it and offer things outside of that. But the core science aspect of it, I think, is what really needs, like, you really need to understand what's going on there to contribute to the scientific body of knowledge, right?
Yeah, that makes a lot of sense. So, for people who, broadly speaking, are considering utilizing like proprietary closed source tools for their biotech or bioinformatics workflows, versus the array of open source tools that are out there, how do you think about the risks of adopting one or the other for something that you're doing?
So when I think about like the trade-offs that you have here, I think it in part depends on the expertise of your team too. So if you're depending on these closed source proprietary tools to perform some very core function of your company, I think that is a risk that you're assuming, because you're assuming that those tools are doing exactly what you expect them to do without actually knowing how they do it. So you lose the explainability of what those tools are doing. That being said, it also means that you can, I guess, push some of that risk onto, say, the company that's licensing you the tool, uh, depending on the licensing terms and everything that you have with them and stuff. So I think for example, in regulatory restricted labs, like having that vendor responsibility makes a lot of sense. In a lot of the research and development side of things, though, most of those use cases are not, the reliability of those tools is not covered under those kind of licenses. And so you really are assuming the risk for the data that comes out of those, and the availability of that tool into the future. If you have a lot of expertise on your team in that particular technology, though, then I would say that the open source side of things, when it's available, is really a good option. If you have the expertise to implement those tools, the expertise to actually dive into those tools and understand what's going on, then I think that just adds a lot of value to the data that you're getting from the tools, and a lot of explainability when things go wrong inevitably, and can help you with that troubleshooting and understanding all the edge cases in your data that you need to model.
Yeah, that makes a lot of sense. And also with adopting open source tools, you have sort of the ability to accelerate the work that you're doing internally, which, when you're a startup and you're resource constrained, is a good place to be, especially when you don't have to pay for the open source alternative to the proprietary thing. So, sort of the trade-offs just make a lot of sense. Whereas when you're later on and you want someone else bearing the liability and maybe cost is less of a concern, it just kind of makes sense to adopt the proprietary tool at that point. So as we head to the end of our conversation, I'm just interested in, where do you see, your work in computational biology, or, the computational biology field more broadly going over the next, say, 3 to 5 years? And how do you see the work that Talus Bio is doing, going into shaping that future?
There are so many exciting things going on in computational biology, and machine learning and AI in biology in particular, I think, are just really exploding with popularity and just the advancements that we have there. One of the things that I think has just traditionally been a fundamental limit of what we can do with computation in biology has been the size of the data that we've been able to collect. I think there's a lot of new technologies that are making that easier and faster and cheaper to acquire lots and lots of high quality data. And I think as the more and more of those come online and we have these larger data collection efforts, we're just going to see more and more of uh, these foundation models and large models specifically tailored for biology come out and be very useful in a lot of domains. I'm really excited for like the synthetic biology angles that a lot of this unlocks. I think we're going to see a lot of the protein design fields really exploding as well, if they're not already. But when I think about like Talus's role in this, I think that our kind of our unique data that we collect, our platform is unique in the type of data that we collect and complementary to a lot of the other approaches. So I think it's really valuable for as like one an integration into that, but then also when we have models that essentially speak the language of transcription factors, then that can also be very useful for understanding, like modeling a cell, for instance, and understanding, when you perturb a particular transcription factor, what's the cascading effects that you have um, for the cell as a whole, instead of just the little bit of the cell that we're looking at in isolation currently. So I think there's a lot of cool modeling that will be happening uh, kind of at those secondary, tertiary, farther down effects of what we can observe and model. So
Yeah, it just occurs to me as you're talking that Talus and its broad characterization of sort of a narrowly focus of its narrow focus on transcription factors, and your vision for understanding the broader biology. There are sort of platform companies being developed with all of these different angles on the cell, and kind of an opportunity for that knowledge to combine itself into a picture that creates this much more nuanced and complex view of the biological phenomena that then would have been possible in the absence of any one of these angles. Am I thinking about that right?
exactly, I think there's all these very cool technologies that are being developed and have been deployed in various ways that uh, combined could really unlock a lot of interesting biology and push forward human health in a really good way. So I'm really excited for the future and what's going to happen over the next few years.
Awesome. Well, Will, it's been a pleasure to have you on. As we head toward the end of our conversation, for people who are interested in learning more about Talus Bio, where should they start if they want to learn more?
Yeah, so we have our website, talus.bio, and the company is pretty active on LinkedIn, and we have a Blue Sky account as well. You can find me on LinkedIn and Blue Sky as well if you want to connect.
Awesome. Well, thanks so much for joining. I really appreciate everything you've shared today, and look forward to connecting down the line.
Yeah, thank you, Ross. I appreciate it.
And that's it for this episode of Data in Biotech. If you enjoyed the episode, please subscribe, rate, or leave a review in your podcast platform of choice. See you next time.